half of the population in the United States actually have some sort of gum disease.
One of the worst outcomes is tooth loss. If you have inflammation which is too severe for too long, then you end up losing the structure and you can lose your teeth.
So what our lab is focusing on is trying to identify the factors that lead to the disease process and ways that we could try to help people out. So our research is focused on finding the inflammatory processes that ultimately can lead to tooth loss, maybe implant problems and those kinds of things. So we work with the immune system, in which we look for factors that trigger inflammation in these cells and try to find ways to tone down that inflammation.
So the ultimate goal of our research is to identify compounds, primarily microRNAs, which can be used to generate novel therapeutics to treat chronic inflammation.
It is important because these cells are the key mediators of inflammation in various diseases, and because our focus is oral cavity, we focus on periodontitis, which is one of the leading causes of inflammation in adults over 30 years of age.
The particular advantage of microRNAs is firstly the profound effects it has on various cell types in various ways. So unlike traditional drugs which target a single pathway or even a single molecule within a cell, the microRNA can target tens or even hundreds of pathways or molecules.
So we want to identify two things from this research. First, whether these microRNAs can be used as biomarkers. The second thing that we want to explore is to use these microRNAs as therapeutics. So the long-run plan is to see if these microRNAs can be used in a topical manner to reduce inflammation. So standardizing some delivery method of microRNAs to apply so that they can reach their target cells and reduce their function to a level that you would see less inflammation and less destruction of the tissue.
One of the biggest advantages in doing my work here at UIC, I would have to say, is the location, and by that I mean the diversity of the population which is around us. In terms of reducing inflammation, if I can demonstrate that effect on a sample which has a lot of diversity, then it's far more likely to translate into a real-world therapeutic effect.
The impact it can have on society and in health in general is that these may be new therapeutics, new therapeutic tools, new diagnostic tools to try to help our practitioners try to control the inflammatory processes. So anything we could do as a clinician and as a scientist to help our patients overcome these challenges actually is very beneficial. So I think our work is actually very innovative in that way.